A very good day, my dear postgraduate students. The topic on the screen is semen analysis, and it is of paramount importance to us, both from the examination as well as the practice point of view as a pathologist. Always think of the subheadings. The subheadings will take us to the answer. Now, I would like you people to kindly be slow and pertinent and see whether you can answer each and every one of these components. For example, the indications, immunological, etc. This kind of pictures we shall be seeing throughout, and they will be helping us. The composition and its significance. This is a very important chart that you will have to write in the exam. You find that there is a component, the source, and then the volume or weight. The bulk of it comes from the seminal vesicle or the prostate, 90%. So this is important for the bulk of it and indicates an obstruction also. The others will be the spermatozoa, epididymis, vas deferens, and the Cooper's glands. And for the examination, we will have to concentrate on the number of spermatozoa and their production, as well as the volume by the accessory glands. This is a histology of the male reproductive system, partly at least the testis, the seminiferous tubule separated by the septate and emptying into the epididymis, within which I am finding the vas deferens and then it empties. So this will be important for us. This histology, you can superimpose this particular diagram onto male infertility. And coming to the schematic histology as such, I find that these are the Sartori cells, the large round polygonal cells. And for every three of them, there will be the reproductive cells that are there, such as the primary spermatocyte, secondary spermatocyte, etc. And ultimately, you find that the spermatozoa derive their nutrition from the Sartori cells. This indicates a mitosis. Now, what are the indications for the semen analysis? So, you find it can be in a case of anxiousness to conceive or anxiety to conceive, male or female infertility. Assess the response to treatment. Effectiveness of vasectomy. Sometimes you find that there is a pregnancy after vasectomy, medico-legal implications. Selection of donor for artificial insemination. Assisted in vitro fertilization. Part of the hormone assay, again medico-legal in the case of rape or in assuming or confirming the paternity. And socio-legally, you find the semen analysis is an important ground for infertility and divorce also. Therefore, it has to be confirmed in the court of law. And these are the series of maturity, but that is not what I am intending this to. You find that the Sartori cells are very snugly opposed to each other, and in between that, there is the spermatogenesis that is happening. Now, what happens is only when these are released to the exterior, there is the production of the reaction by the antibodies. The mode of collection, obviously a private room adjacent to the laboratory, not from a distance. And for in vitro fertilization, motile sperms will have to be separated. There is something called as a seminal plasma. So this is a sample over here. There will be a supernatant plasma, and after centrifugation, there will be the spermatozoa. The plasma itself is important for biochemical and antibody study. Silastic condom is used during the intercourse, and this was designed by this person, Dow Corning, 1948. No partial sampling is allowed. Obviously, two labs will give two different values, leading to legal problems. And on examination, we'll have to look at the volume, usually about 1.5 ml, and it is gray-white, opaque. Sometimes it will turn yellowish. The viscosity, of course, it is viscous, but then it has to liquefy. More than 2 centimeters of the length of the thread indicates increased viscosity. The count obviously will have to be made, the morphology has to be entered, the motility of the spermatozoa, and the vitality. More than 58% should be vital. And this is a picture of an artificial insemination. You find that after all the preparations, it is being injected into the female genital tract. Artificial insemination is different. In vitro fertilization is totally different. The physical examination, this is what I said. Based on the color and the clearance, you find it can be clear or watery, 2 is cloudy, 3 is milky, 4 is creamy. Sometimes you find that it can be yellowish or yellowish red because of an admixture with the blood or the feces. This is a very important slide for you people, and for in vitro fertilization, it is important. So there is a medication and ovarian stimulation over here, after which you find that you will have to track the development of the ovarian follicles. There has to be a continuous monitoring that is there, and there is a retrieval of the egg that is there. And there is a transfer, and there is also a study of the embryogenesis in culture. Finally, there is a transfer of the embryo and it is implanted. The development is being studied. Finally, the pregnancy is being confirmed. So this particular cycle is extremely important. I have made it as easy as possible. Examination of the wet count of the sample is again important. Incubated at 37 degrees for liquefaction. 30 to 60 minutes is the maximum time. Volume, again I have mentioned, pH has to be 7.2. A lower pH indicates acidity. And the wet preparation is used for testing the motility. Here again, beautifully you will be able to see the motility of the sperms. And diluting is used for counting, just like any of the RBC or the WBC count. And these are tests for the immunology, immobilization test, immunobead test, or the MAR test. You people will have to mention all these things. The sperm motility and the grading. So this is the grading and you people will have to memorize it. 0 is no movement, 1 is no forward progression, it just moves something like a Brownian movement, and 2 is there is a slow forward progression, and 3 is slower speed, 3B, and there is a lateral movement. Finally, there is a rapid movement. We used to call it as a darting motility, as in the case of Vibrio call. So the instructions to the patient are these. Avoid ejaculation, it has to be, abstinence has to be there for at least about 72 hours. And avoid the drugs, caffeine, etc. Of course, we will not be taking marijuana, etc. But these are important. Any herbal medications and hormonal therapy. All these should be avoided before the test. The temperature I have already mentioned, and the sperm motility, these are the concentration samples which you will have to read. This will have to be diluted with the seminal plasma. Centrifugation is done at the rate of 1500 RPM for a period of about 6 minutes, and then the semen is being separated, or the semen plasma is being separated. From the dual chamber, you will be able to do the counting as it is shown over here. Now, this is the age-old method of counting. The RBCs will be done in the middle, the WBCs over here. And it is these 4 squares that are used for the semen analysis. Basically, there are 2 types of chambers. One is the improved Newbar chamber. I'm not going into the details of it, but then there is another chamber called the Fuchs Rosenthal chamber. Normally, you find that there will be a depth of about 1 millimeter only in the case of the Newbar chamber, whereas it can be 0.2 millimeter, 0.1 millimeter here, and 0.2 millimeters deep. Therefore, in a lower count always, you find that the Fuchs Rosenthal chamber, for example, in a cerebrospinal fluid, can be done. So this is a formula for the counting. I would like you people to kindly go through. There is a charging and the humid conditions are there. The large squares are being counted. This formula, a little difficult, but try to memorize it. I'm going off track over here. Now, there's not a sample of semen, but then a patient was asked to give us urine. He brought it something like this, and then they said it should not be a semen, and again you'll have to bring it. He again brought it, again it was appearing milky white. It is a case of chyluria, where the lymph is there, similar to the semen in appearance. Automation can be done just as in the other cell counts. By computer-aided methods, fluorescent for DNA analysis, tail detection by algorithm, and measuring the motility and the sperm concentration. And according to this, at least 200 sperms will have to be studied. And these are all linked to the computer software for data. That is what over here. Computer-aided semen analysis. The uses will be this. There is a fluorescence, concentration, motility, morphometry can be studied, it can be measured, reproducible images. That is again one of the advantage of the computer. And the important parts of it are being studied as well as the motility. Advantages are quick analysis and it is reproducible. Highly detailed analysis is possible and there will not be manual error. And greater statistical power is there. You will be able to compare also. This is something that is happening in automation. You find earlier we used to look at it under the microscope and then take a picture of it. Now there can be an attached one with this. So you find that there is a machine just like your cell count, a Sysmex or so, and you find that there can be a picture that is over there, the count is being recorded on the computer. There are certain parameters that you will have to see in the sperm. And this is one story of artificial intelligence. So the machine, what does it do? The concentration, motility, morphology, any of the DNA damage is also being studied, and also the method of sperm extraction. Everything is being given over here. So when there is going to be an artificial intelligence replacing the mechanical methods, you find that we are digging our own grave. There will be a day when pathologists will no longer be required, and not more than 10% of us will be needed for any of the hospitals. So these are the strategies. This again is important. This of course is a biological study which has been imposed in the human beings. One will be the study for the meiosis. So I made it again simple over here thanks to Cell and Tissue Research Kit. There is a meiosis in which you'll be able to study these three things: molecular markers and epigenetics, and R.A.T.S, assisted reproductive technology. So in which you will be able to study these things. And finally, in vivo, you will be able to study the histology, the semen production, and the fertility restoration upon re-injection into the animal. Also, the changes subject to irradiation. So please do remember these three things: meiosis, R.A.T.S, and in vivo testing. What are the pitfalls in the semen analysis? So this again is a very important one, very easy. So there can be defects in the sample collection, sperm concentration, in the motility, in the morphology. So these are all important and manual errors can be there. Therefore, a double check is important. And checking for the abstinence, for example, ID, almost all the things that you people follow in your automated hematology laboratory can be followed over here. Motility and the other things, these should be checked and signed by the competent pathologist. Anti-sperm antibodies are important. As I had been mentioning earlier, there is usually a tight junction between the Sartori cells, but once the spermatozoa are being released to the exterior, there is a production of antibodies. There is a story of mumps. There will be the sperm immobilizing antibodies, but it becomes active only when the patient attains the reproductive age group. So they impede the motility. Usually it is the IgG or the IgM. They can also impede the interaction with the oocyte. And they are found after all these procedures: vasectomy, testicular biopsy, vasoepididymyostomy, varicocele, or torsion of the testis or womb. All this can lead to the production of anti-sperm antibodies. See, this is what is happening. So there is a antibody over here, and it is again coated with it. The latex bead is coated. There are the antibodies already attached to the spermatozoa. Therefore, this latex bead test will make us make a diagnosis. Even otherwise, you will be the clumping, see the clumping as such. So there are the cross-linking anti-human antibodies over here, the anti-sperm antibodies over here, and there is a antibody that is bound to the latex. So it is something like a sandwich technique that you people see in immunohistochemistry. Thanks to this wonderful reference. And this incidentally means total absence of sperms or total clumping. See this, when I say clumping, it can be head-to-head clumping, or it can be head-to-tail clumping, tail-to-tail clumping. Finally, it is being counted as the percentage, which is again graded as this. This you will have to mention. No adhesion or clumping, and this here it is mild, moderate, and severe or large. The general findings are these. So you'll have to report for normal spermia, normal number of sperms, oligo, and then asthenospermia. This is the number of patients usually, a data that has been produced by the WHO. The fructose test is important. It is mainly indicated for the seminal vesicle, and an absence of fructose indicates that there is an obstruction. So the fructose test is positive and it is negative. MAR test is again over here. It is a semi-quantitative, non-automated diagnostic kit for identifying the anti-sperm antibodies. As I told you, it can be IgA or IgG. The morphology. In fact, this should have been one of the first of the stories. You find there is the morphology of the spermatozoa. You find that there is a sperm head which has got somewhat a conical contour, and then it is covered by an acrosomal cap. There is a nucleus, nuclear vacuoles are there, and this is the mid piece, and this is the end piece of the tail finally. So the mid piece is over here. This will be containing mostly the mitochondria, and ultimately you find there is a terminal disc. Externally, there is a cell membrane. So this is an important one which you people shall remember. And hats off to this person, scientist who draws. So this hat is meant for him. The morphology we people should. The morphology. This is what you people will have to follow. This is a normal sperm. It can be double-headed. There can be a giant head. Sometimes it is amorphous, the pinhead or small one. There is a tapered head, constricted one. Just like as even the sperm can have so many of these abnormalities. And finally, there is a head without a body and soul. So this is an incidence of the sperm, and it can be identified by numerous stains. Please remember this. Eosin nigrosin is one, and for the morphology, the PAP, rose Bengal, Toledan group are all ones that can be used. So this is the way you people report, whether it is alkaline and what is the count over here, what are the actively motile ones, non-motile, sluggishly motile, the normal forms. Here in this case, it is about 95%. And ultimately, this will be the normal values that are given by the WHO. The sperm volume, concentration, count, progressive motility, and normal morphology more than 4% would suffice. Non-sperm cells are also there, but they should be less than 1 million. Otherwise, they might indicate infection. So azoospermia, this can be one of the causes. The beautiful diagram by the Cleveland Clinic. Here again, the male reproductive tract, it can be recalled. The testes, and then you have got the epididymis, the vas deferens, etc., the penis over here, and then the other glands. So all these are important. Seminal vesicle, for example, as well as the prostate. A normal count, oligospermia and azoospermias. So these are the various parameters, and this can be a checklist for us. See whether all these are being followed. It's a beautiful summary thanks to Springer link. So the appearance, odor, viscosity, volume, agglutination, concentration, motility, morphology, then vitality of the sperms as well as the round cells. Thank you for your patience.